Today's lecture is part 2 of a several lecture series dealing specifically with the adaptive immune response. In the previous lecture, we talked about things such as immunogenicity, what makes an antigen a very good or strong and effective antigen, and then we discussed a little bit about the differentiation process that occurs with both B cells and T cells in their activation in order to produce effector cells and memory cells. We also then began a little more cohesive discussion on B cells and antibodies and antibody structure. So today we're going to continue that lecture topic dealing specifically with B cells and antibodies that are produced by B cells. The first note that we want to discuss is antibody-mediated effector functions. That is, what exactly are these antibodies doing that are being produced by the B cells? Oftentimes, in general terms, what we typically say antibodies do is that they target or tag a pathogen for ultimate destruction. However, we know there is some fine-tuning associated with those antibody-mediated functions. That is, again, what the antibodies that are being produced are doing. This slide gives you an overall summary of the topics that we're going to discuss, which include neutralization effect of antibodies, how antibodies act as opsonins, how antibodies activate the complement system, this third facet that we have alluded to a few times throughout the previous lectures, and we will eventually have a full-blown lecture dealing with the complement system, and then a mechanism known as ADCC, which is short for antibody-dependent cell-mediated cytotoxicity. The first antibody-mediated function that I want to focus on is neutralization. The term neutralize means to stop someone or something from being effective or harmful. And in this case, antibodies that have a neutralizing function do exactly that. They stop something, the potential pathogen, from being effectful or harmful. And this slide does a really good job of giving us a summarizing view of what that neutralization looks like. In the top portion of the figure, you have an example of toxins that are being produced by a potential pathogen. And again, we discussed that toxins are molecules that are produced by pathogens that help with their virulence and their infectious process. In this particular case, what you see is the toxin molecules in the top portion of the panel interacting with the cell in question and causing cell damage overall. However, in the second portion of that top panel, you can see that they have the toxin molecules. However, in this case, there is antibody production that will be able to bind to the toxin in question. If, in fact, that antibody is being produced, binds to the toxin in question, it binds up the toxin and prevents the toxin from doing the damage that it would normally do to the cell, as we saw in the top portion of this particular figure. Another example of neutralizing capability is in the bottom portion of this particular figure. In this case, we have a virus that has the potential of binding to and interacting and infecting red blood cells. However, if there is an antibody that has the ability to bind that particular virus, it binds the virus and prevents it from actually entering into the host cell, which in this case is a red blood cell. So in both of these examples, we have antibodies that are specific for the antigen in question. They bind to that antigen and prevent the antigen from doing the negative or deleterious effect that the antigen would normally have. With this slide, we are focusing in a little more detail on viral pathogens. I have mentioned previously that what has to happen with a virus is that it needs to infect a host cell, and it takes over the host replication, transcription, and translation machinery to make more viruses. And it turns out that antibodies can, in fact, act as neutralizers in many different cases or examples with viruses. In this slide, again, we're looking at several ways that antibodies can neutralize the effect of a virus. In panel A, what we are simply looking at is blocking the receptor engagement of the virus. So in this particular case, the antibodies bind to the virus and prevent the virus from actually interacting with the receptor that it needs to interact with in order to gain entry into the cell. This is obviously the best location to prevent viral infection, is to block the entry altogether. However, we also know there are other examples of blocking viral entry. In panel B, what we're seeing is antibodies binding specifically to the receptor that the virus would interact with in order to gain entry. In both panel A and panel B, the entry of the virus is being inhibited just in two different ways by antibody binding. Panel A, the antibody binding to the virus itself. Panel B, the antibody binding to the receptor, preventing the virus from binding. In panel C, what we see is a antibody interaction with the virus. However, in this particular case, this does not prevent the virus from entering into the host cell. What this particular situation does is continue to bind to the virus once it has entered into the cell, but then prevent the unpackaging step that takes place with viruses in order to get the genetic material out and into the host system. In panel D, you see antibodies binding to the virus externally before the virus actually gains entry into the host cell. However, in this particular case, entry is blocked in a different way than what we discussed in panel A and panel B. In this particular case, the virus, even though it can interact with the receptor, can be drawn in by the receptor, lacks the ability to be able to be released inside of the host system. So lastly, in panel E, what we're looking at is a situation where let's assume that the virus did make it in, did actually replicate. In panel E, what we have are antibodies that bind to the virus and prevent the release of that virus from the host cell. So there have been antibodies detected in all of these different stages that have the ability to neutralize the effect of a virus, just in different stages of the viral infectious process. Another antibody-mediated effector function is known as opsonization. So this is a term that we have encountered a few times previously. Opsonization basically promotes phagocytosis of antigens by typically macrophages and neutrophils. So an opsonin is a molecule that binds to a potential pathogen, targeting it for phagocytosis. In this particular figure, you see two different examples of what is happening with antibodies acting as opsonins. If you recall, in our last lecture, when we talked about antibody structure, there were several components of a typical antibody that we focused on. We talked about the Fc portion, which is what I equate to the stem of the Y, and then the Fab portion, which is the arms of the Y, where the specific interaction between antigen takes place. So in the left-hand panel, what you're looking at is a very straightforward example of a macrophage in yellow, right in the center. And associated with the macrophage in the blue is what's known as an FCR. This is an Fc receptor, meaning it's a receptor that is found on the surface of the macrophage that binds to the Fc comportion of an antibody. Above the Fc receptor in this figure, you see in pink the pathogen that is being bound by antibodies. And again, it is those Fab portion of the antibodies, the arms of the Y, that are specifically binding to the epitope of the antigen. And also in this figure, you can see the Fc portion interacting with the Fc receptor on the macrophage. This is a clear example of opsonization in that the antibodies are binding to the antigen in question, tagging it as an opsonin, and marking it for phagocytosis. And this is exactly what happens because of this complex interaction. On the right-hand side is a much older figure, but also I think does a very nice job of depicting the interaction that is taking place in this complex way. If we start at the top of the figure, we have a rather large-scale pathogen in green with all the little knobs extending off of the surface. And what you can see is the antibodies are interacting with the antigen. Again, the Fab portion of the antibody interacting with the antigen. The stem of the Y, or the Fc portion, is bound to the Fc receptor, which is in red, and the Fc receptor is part of the cell membrane of the leukocyte in question, macrophage, neutrophils, for example. In this very complex interaction, this then triggers that receptor ligand interaction and the signal transduction that we have talked about multiple times already throughout this first week of material. That interaction causes a signal transduction pathway to ensue, and that then causes cellular responses to take place. And those cellular responses in this specific case are to trigger phagocytosis, the taking in of the pathogen and then the overall destruction of that pathogen. The next antibody-mediated effector function of antibodies is to activate and assist in the complement system. So again, I've mentioned the complement system a few times in the previous lectures, and the complement system is in fact a third facet of the immune response, which we will spend an entire lecture talking about. So for now, what we want to just discuss is that these antibodies do in fact play a role in activating and assisting the mechanism overall. In the left-hand portion, what you can see is one aspect of what happens as an ultimate result of activating the complement system is antibodies interact with the antigens in a target cell. In this particular figure, that's listed as a tumor cell. Above the antibody, you see this complex set of molecules that includes C1s, C1q, C1r, and these are molecules that are part of the complement system that recognize that antibody-antigen interaction and trigger a very complex mechanism known as the formation of the membrane attack complex, or MAC. This formation of the membrane attack complex plays a role in destroying the membrane of the cell in question and hence causing the destruction of the cell in question. This is specifically a part of the complement system and occurs because of the activation of antibody-antigen interaction. On the right-hand side is another aspect of that activation and assisting in the complement system. What you see is an antibody interacting with the pathogen in question. This then also triggers this activation of the complement system, which ultimately results in the production of a molecule known as C3b, and C3b, in conjunction with the antibodies, help target the pathogen for phagocytosis. So both of the examples on this particular slide are specific complement system functions, but the point here is that in order to activate it, there needs to be recognition of antibody-antigen complexes by a macromolecule complex that you see in the left-hand panel with C1, C1s, C1q, C1r, known as the C1 macromolecule complex. The final antibody-mediated effector function that I want to discuss is a mechanism that is abbreviated ADCC, which is short for antibody-dependent cell-mediated cytotoxicity, which is rather a mouthful, hence the abbreviation ADCC. With this slide, you see two summarizations of what is happening during ADCC. Let's just go back to the title overall. Antibody dependent, meaning antibodies are going to be involved in this mechanism, and cell-mediated cytotoxicity. So in this process, antibodies are going to help with the targeting of destruction of self cells. Now, I have used this term, I think, previously in a few lectures, and that oftentimes we have immune cells that interact with altered self cells. Altered self cells is a very generic term, but oftentimes it refers to cells that are either infected with the virus or cells that have gone out of control and are part of a tumor formation. So these are self cells that need to be destroyed for one reason or another. They've gone out of control or they've been infected with the virus. And in this specific mechanism, we have natural killer cells that are involved in the process. So again, both figures are depicting the same concept. On the left-hand figure, you have a natural killer cell. In the blue is the Fc receptor that's part of the natural killer cell. Just like in the previous example, we know the Fc receptor binds to the Fc portion of an antibody. In this figure, you also have antibodies binding to either an infected cell or a tumor cell, infected with virus or a cell that is part of a tumor. And in this particular case, there's this complex interaction and recognition of antibody with the Fc receptor and the natural killer cell and the cell that needs to be targeted for destruction. When this complex interaction occurs, the cell in question, the target cell in question, is in fact targeted for destruction, and we'll talk about that in the next slide. So the right-hand panel shows exactly the same thing with just a different figure. Coming in on the right is the antibody interacting with an infected cell or tagging an infected cell. You see at the bottom portion of this figure the natural killer cell and the Fc receptor, in this case in yellow. The Fc receptor, part of the natural killer cell, binds to the Fc portion of the antibody that is bound to the infected cell or tumor cell. Once this complex interaction takes place, the natural killer cell does what its name implies, is kill the cell in question and targets it for destruction. So in this figure, we're looking at this ADCC mechanism in a little more detail, going from panel to panel. So in the far left, we have just what we discussed in the previous slide, antibody binding to antigens on the surface of the target cell. The target cell in this case is either an infected cell or a tumor cell. In the next panel, you can see that the Fc receptors from the natural killer cells recognize the bound antibody, and you have this complex interaction again that we discussed on the previous slide. This complex interaction then triggers the natural killer cells to produce and release molecules that are going to play a role in the destruction of the target cell. The natural killer cells release a number of different types of components or molecules. They release enzymes known as granzymes, and granzymes are general enzymes that help degrade proteins. There are other proteases, general class of enzymes that degrade proteins that are released. There are also molecules known as perforin, and these are molecules that perforate the cell membrane of the target cell, which then allow for these other molecules that are helping with the destruction of the target cell to take place. This mechanism of destruction is a very well-defined mechanism known as programmed cell death, and more specifically titled apoptosis. Apoptosis is a mechanism that has very characteristic features associated with the death of the cell, and so oftentimes scientists can very easily recognize a cell that has been induced by this mechanism of apoptosis because of the clear structural features associated with the cell and the clear mechanism of destruction that is taking place as well. The last bit of information that I want to talk about for the remainder of this particular lecture is dealing with the fact that antibodies are not just one size fits all. It turns out that there are 5 distinct classes of antibodies, or as we discussed in the previous lecture, immunoglobulins. There are, in fact, 5 distinct classes, and what we'll do is we'll discuss them each in a little bit of detail. So the first one on this particular slide is immunoglobulin G, also abbreviated IgG. It turns out that this actually is the most abundant class of antibodies that does in fact get produced. It usually plays a very big role in opsonization. Again, that process or mechanism of enhancing or triggering phagocytosis. There are 4 distinct subclasses associated with IgG. We're not going to look at individual functions of the subclasses, but they are listed here, and I just want to make sure that you are aware that there are, in fact, 4 distinct subclasses. This particular antibody class typically plays a really big role in fighting off bacterial infections, but is also known to be elicited against fungal infections as well as viral infections. And this particular class of antibody is actually the only type known to cross over the placenta, so that an immune response does in fact get transferred from the mother to a developing fetus during the gestation period overall. The next antibody class I want to talk about is IgM. IgM makes up about 5 to 10% of total serum immunoglobulin. It turns out that this particular class of antibody is large and it doesn't diffuse well, and that's because the true functional form of the antibody is a pentamer function. So you have 5 individual IgMs that come together to produce this large pentameric structure overall. It has been determined that this tends to be the first class of antibody that is produced in response to an antigen. It does play a huge role in activating the complement system overall. And again, going back to its really large size, it's not found in very high concentrations in intracellular tissues because it's very hard for this molecule to diffuse out of the bloodstream into the tissue and then from the tissue back into the lymph. So this particular class, again, is not typically found in very high concentrations in intracellular tissues. The next class of antibody I want to discuss is IgA. IgA is very effective in neutralizing capability. This is the predominant class found in secretions. So it is found in breast milk, saliva, tears, mucus, and it is considered the secretory Ig molecule. That is, when it is produced, it is secreted into these components that I had just discussed. Production tends to be very high of this particular class of antibody. It makes up about 10 to 15% of total immunoglobulin in the serum. The job of this particular antibody is to protect body surfaces that are exposed to the outside or external components, so that foreign substances can, in fact, not enter into the host system. This particular antibody, similar to what we just discussed with IgM, in its functional state is actually a dimer, meaning 2 individual IgAs come together to form the functional form of the IgA antibody. The next class of antibody is IgE, and IgE's job is to typically play a role against parasitic infections or play a role in the immunity to parasites overall. However, it turns out that this particular class of antibodies also plays a really big role in hypersensitivity reactions. In the first lecture, we had discussed briefly the fact that what we would do is spend a lot of time learning about how the immune system functions, and then once we have a good handle on that, we will talk about dysfunction when the immune system doesn't function properly. One class of problem associated with the immune response is hypersensitivity, which we oftentimes just refer to as an allergy. IgE plays a really big role in one subset of hypersensitivity reactions, which for now we're going to just say that it does play a role in these allergic reactions, and we'll discuss the details of that when we get to hypersensitivity overall. The fifth and last class of antibody is known as IgD, and IgD is found in only very small amounts in serum. It turns out because there's so little of this particular antibody found, we don't necessarily know the entire function overall for IgD. It is thought to play a role in activating B cells, and so that this is a helper or an accessory type of antibody to help the activation of B cells like we discussed previously. It is in fact a major membrane-bound immunoglobulin, and it is in fact expressed by other mature B cells. But again, in this case, we don't have a lot of information on the exact function of this particular class, other than it's thought to play a role in activating B cells overall. Lastly, I want to mention that antibodies can be categorized in a number of different ways. The first categorization is something known as an isotype. An isotype, if you look in panel A, highlighted in the black, shows where differences may occur from one molecule to the other. These differences occur in the constant region of the antibody, and this is specifically what allows or distinguishes between the classes or the subclasses. So in this example, what differentiates an IgG from an IgM or from an IgA is the constant region. The sequence that's found there is slightly different and is characteristic of either IgG, IgM, and so on. The next category in panel B is allotypic determinants, and an allotype is really a variant that is encoded by different alleles. So when we have allelic differences, this is what refers to an allotypic determinant. So in this example, again, you can see sort of highlighted with the hashtags are where the sequence differences occur. In the specific example, you have two IgG class 1 molecules, but they are slightly different from one another in one strain or another. And this is again due specifically to allelic differences. Functionally, they're all IgG class 1, but there are subtle or slight differences due to different alleles. And then the last categorization is idiotypic determinants or idiotype. And the idiotype differentiates at the interaction or the antigenic determinant portion. So in the generic figure in panel C, you can see the black highlights that Fab portion, but more specifically the part that interacts with the antigen. This is the variable region, and this does vary from antibody to antibody to antibody to allow for all of the specificity that we've discussed previously. So in the example that you see here, we have two different IgG class 1 molecules. They are categorized as IgG class 1. However, the antigenic determinant or the binding portion is different, and so they are in fact different antibodies binding to different antigens.